Welcome to Lecture 19, where we will go over chromosomes and linked genes. Chromosomes are considered to be the base physical unit of inheritance. This is what is passed on from parent to offspring. The locus refers to the location on a chromosome where a gene is actually located. Mendel's law of segregation and independent assortment occur because chromosomes separate out and also assort themselves independently in meiosis. Remember that both of these laws occur in anaphase 1. Law of segregation refers to when those homologous chromosomes separate from each other, and the law of independent assortment refers to the fact that all of those tetrads will assort themselves independently within the cell, such that when producing gametes, it is completely random as to which side the homologous chromosomes will separate to. Sex linkage refers to genes that are located on the sex chromosomes. They can be linked to either the Y or X chromosomes. However, most of the sex-linked genes will be linked to the X chromosome. It was first studied in fruit flies by Thomas Hunt Morgan. The genome of the fruit flies resembles humans such that the sex determination for females occur when the organism has two X chromosomes, and the males are determined by having an X and a Y chromosome. In total, they have 4 chromosomes, 3 autosomes, and the sex chromosomes. When studying sex linkage in flies, we can term the alleles as wild type or mutant. Wild type alleles refer to the alleles that are considered to be normal and are most common in the population, whereas the mutant alleles are considered to be more rare. Mutant alleles typically involve a loss of function, but this is not always the case. An example of these alleles are eye color in fruit flies. White eyes are considered to be the mutant allele and occur because there is a lack of pigment production. We would refer to the white-eyed allele as simply a small w. Red eyes, however, are considered to be the wild-type allele. In this case, we would refer to that allele as W+. This slide depicts Thomas Hunt Morgan's experiments. In his parental generation, he took red-eyed females and crossed them with white-eyed males. He noted that all of the offspring, both male and female, in his F1 generation had red eyes. When he took males and females from the F generation and crossed them to obtain his F2 generation, he noted that all the females in the F2 generation had red eyes, whereas half of the males had red eyes and the other half had white eyes in the F2 generation. Let's take a look at the genetics of what is going on in these crosses. The P generation consisted of a true wild-type female, such that she had 2 X chromosomes, both carrying the wild-type allele. The male in this cross was a white-eyed male. As a result, this male would have an X chromosome with a mutant allele, as well as a Y chromosome. If we take a look at the gametes that could be created from the parental generation, we would note that the female could only produce one type of gamete. All of her gametes would have an X chromosome with the wild-type allele. The male in this cross, however, would have 2 potential gamete types. Half of them would contain the X chromosome with the mutant allele, and the other half of the gametes would contain the Y chromosome. The resultant F1 generation, thus, would have females with 2 X chromosomes, one with the wild-type allele that they inherited from the mother, and the other X chromosome would contain the mutant allele that they inherited from the father. The males in the F1 generation would have automatically obtained the Y chromosome from their father. As a result, they would have obtained an X chromosome with the wild-type allele from the mother. In both instances, both males and females would exhibit the red-eyed phenotype. If we take a look at the F2 generation, we have to consider what kind of gametes the F1 generation could produce. The females in this case have 2 potential gametes. Half of their gametes would have the X chromosome with the wild-type allele, and the other half of the gametes would contain the X chromosome with the mutant allele. The F1 generation males would produce 2 different gametes. Half of their gametes would contain the X chromosome with the wild-type allele, and the other half would contain the Y chromosome. If we take a look at the resultant F2 generation, we see that the females in the F2 generation have 2 genetic possibilities. Half of these females would carry the X chromosome both with the wild-type allele. The other half of these females would be carriers. They would contain an X chromosome with the wild-type allele and another X chromosome with a mutant allele. The males in the population would also have two different genetic possibilities. Half of the males would have the X chromosome with the wild type allele and the Y chromosome, and the other half would carry the X chromosome with the mutant allele and the Y chromosome. As a result, we can see that all of the females in the F2 generation would have red eyes. However, half of the males in the F2 generation would have red eyes, and the other half would have white eyes. In humans, we typically denote females as having an XX genetic makeup and XY for males. Please note that there are many other circumstances that can actually alter gender determination. However, for the purposes of this course, we are simply looking at XX and XY to understand linkage on the sex chromosomes. As we can see when we look at sex determination, it is the chromosome that is present in the sperm that will determine the overall gender or sex of the embryo. The Y chromosome contains a particular region called the sex determining region or SRY region. On this location of the chromosome, the genes will be located for various traits such as testes function and sperm production. The X chromosome is considered to be essential because it is present in both males and females. The X chromosome can contain many other genes that are unrelated to gender. Some genetic disorders can be carried by the females or carried on the X chromosomes, and they are due to mutant recessive alleles. However, there are some genetic disorders that can be linked to the Y chromosome. There are also some genetic disorders that can be considered dominant versus recessive. When you have these genetic disorders that are due to mutant recessive alleles on the X chromosome, many of these diseases are typically only seen in the male population. Common examples of these types of diseases include hemophilia, muscular dystrophy, as well as red-green color blindness. This slide here depicts a typical sex linkage problem. In this instance, we have a fully healthy female. We denote her as being fully healthy by giving her two X chromosomes with a capital N allele, which signifies that she carries the healthy alleles. On the right-hand side of this diagram, we have a male that is affected by muscular dystrophy. His genetic makeup would be XY, and on the X chromosome, he would carry the mutant allele. In this case, it is denoted with a small n. If these two individuals were to mate, we would see that the only alleles or only gametes that the female could produce would be an X with a healthy allele, capital N. The male would produce half of the gametes with an X chromosome that would carry the mutant allele, and the other half of the gametes would contain the Y. We can see that in this mating, the offspring potential are as follows. The females from this cross would all be considered carrier females. This is because both of them would carry a healthy allele from the mother and the diseased allele from the father. However, phenotypically, these females would be normal. The males from this offspring, the male offspring from this cross, would all be considered normal males. We can see that they obtain the Y chromosome from the father, and they also carry the healthy allele from the mother. Let us now consider two different possibilities for these carrier females. If a carrier female mated with a normal male, we would see the potential for offspring below on the diagram to the left. In this type of mating, the carrier female and normal male would have the following offspring probabilities. With respect to their female offspring, they would have a completely normal female that would carry both healthy alleles. However, half of their females have the potential to become carrier females. With respect to the males from this crossing, half of the males would be normal and the other half would be affected with muscular dystrophy. So we have all four possibilities: completely healthy females, carrier females, healthy males, and affected males. If we take those carrier females from the F1 generation and then mate them with a diseased male, we would see a different scenario. In this case here, we would have a carrier female being produced as well as an affected female. We would also have normal males as well as an affected male. We can see that in this scenario, we have a 50% probability of having offspring that are affected by the disease, such that half of our males would have the disease as well as half of the females. The other half of the females would be considered carriers as well. So we can see that in this genetic cross, we have a higher occurrence of the mutant allele. This diagram here illustrates the pedigree from the royal family. The royal family was considered to have royal blood. However, this royal blood actually referred to the condition of hemophilia. In a typical pedigree, you can denote sex by having the different shapes that represent the different genders. Females are normally represented with a circle, whereas males are normally represented with a square. We can also use different colors to represent healthy versus affected individuals. In this case, a healthy or normal individual would be denoted in yellow, whereas the disease would be denoted in green. In this case, we can also see that there are carrier females within the royal family. We can also see that there is a higher occurrence of males being affected with the disease. In fact, none of the females are truly affected by this disease. However, they are considered to be carriers. In this case, they would have a healthy allele on one X chromosome, whereas the other X chromosome would contain the mutant allele for hemophilia. We note that this is much more prevalent in males because as long as they have one copy of the X chromosome, they will, as long as that copy is mutant, they will express the mutant condition. So in males, it is either they have the healthy allele or they have the diseased allele, and they exhibit that phenotype. In females, there's the ability to either be completely healthy or be a carrier, and even the carriers will not exhibit the disease. When looking at sex chromosomes, as stated earlier, only one X chromosome is necessary for survival. We see this in the condition of males. As a result, in females, you could potentially get the double dose of X genes if both of those X chromosomes were expressed at the same time. As a result, female cells will undergo what is termed X inactivation. In these instances, one of the X chromosomes will be inactivated and will lie dormant within the nucleus. This inactivated X chromosome can be referred to as a Barr body. The choice of which X to activate or inactivate is completely random and usually occurs during embryonic development. Because this occurs early in embryogenesis, when one X chromosome is silent, the resultant cells that would result from cell division or mitosis from that original cell will also have the same X inactivation pattern. This will lead to the creation of mammals that are mosaics. Note that mosaics are only present in females. In some groupings of cells, they will express the maternal X, whereas other groupings of cells will express the paternal X. The pattern of X inactivation is considered to be reversible. This is because during meiosis, the Barr bodies are reactivated to ensure that all gametes get a truly active and healthy X chromosome. This diagram illustrates the concept of X inactivation. In this instance, in the early embryo, we can see that there are two X chromosomes. One carries the parental allele for orange fur and another contains the parental allele for black fur. One would have originated from the mother, whereas the other one would have originated from the father. After a few rounds of cell division, the X chromosome would undergo X inactivation. In some instances, the active X would be the one with black fur, so the black fur traits would be expressed, and the X chromosome for the orange fur would be silenced, and we have vice versa in the other groupings of cells. As a result, we have some groups of cells that will express the maternal coloring and other cells that will express the paternal coloring. This leads to the formation of a mosaic as seen in the diagram below. Some things to remember about X-linked traits is that mutant or rare alleles will only show up in males. As stated previously, these mutant alleles are usually considered to be the recessive allele. In order to truly express the mutant condition, females would need to get 2 copies of the mutant alleles inherited on both of the X chromosomes, whereas males only need to inherit 1 mutant allele. This is because they are hemizygous in their genetic condition. They only carry 1 X chromosome, whereas the other one is a Y. As a result, males will always get the Y chromosome from their father, and they will always be obtaining the X chromosome from their mother. When a diseased father has a daughter, he only has 1 X to give, and if he carries the mutant allele, he would be passing that on to the female offspring.